Good morning, students. I am Dr. Sujata. Today we are going to discuss about bacteriophage. Bacteriophage means the virus which infects the bacteria is called as bacteriophage. We are studying about T4 model. Coming to the introduction, these are bacteriophages are very small acellular particles. They can replicate only in the host. This doesn't have any machinery to replicate. So they always require host machinery for their replication, for their multiplication. They are obligate intracellular parasite. They are always presenting intracellularly. They doesn't present in the externally, that means in the environment. They consist of nucleic acid genome. They consist of only nucleic acids and protein code, which is known as capsid. So first of all, we have to know who discovered this. Bacteriophage was first discovered by Frederick. Frederick is the scientist who first identified, who first recognized there is the bacteriophage is there. Bacteriophage means the virus which eats the bacteria, is called as bacteriophage. This bacteriophage lyses the bacteria, it completely lyses the bacteria. That's why the name is called as bacteriophage. As you might all know, it is an obligate intracellular parasite. It is an intracellular parasite. It doesn't survive in the environment. It doesn't have any replicatory mechanisms. There are at least 12 distinct groups of bacteriophages. A very diverse group are there. Okay. Coming to this, the bacteriophage, you have to remember that what is bacteriophage? Bacteriophage is nothing but the virus which infects the bacteria. It was first discovered by Frederick. After that, in 1950, Clark is also scientist. He also proved that there is a bacteriophage is there. Okay. Next, coming to the structure of this bacteriophage, it consists of a head. Head is present. Head, tail in the region, tail region. Head is icosahedral symmetry. It is an icosahedral symmetry which consists of 53 micrometers long, and it consists of a double-stranded DNA, which we already discussed. It consists of a nuclear genome, that is nothing but double-stranded DNA. The tail is helical symmetry with the core tube. It is surrounded by a protein sheet. This protein sheet is arranged in 24 units of 6 subunits. The sheet is connected to the collar, and its upper and basal parts is at the lower end. The base plate is hexagonal with single tail or spike at each corner. A thin tail filament is present. The head is a prism-like hexagonal having 950 into 650 angstrom in diameter. The tail is 950 angstrom in length, joined to the head and neck and a collar. Okay, now coming to the composition, it consists of only nuclear matter. It may be of DNA or it may be of RNA. All the phases consist of head, tail, connected by the collar. Okay, icosahedral symmetry. Bacteriophage, T4Phages, icosahedral symmetry. Next, coming to this. This is a bacteriophage. We have to remember bacteriophage. It is a virus which infects the bacteria. Here you can see the head, tail fibers, collar, head sheet, and the hexagonal head will be there. Tail fragments will be there. This is the model, T4 model of bacteriophage. Next, coming to the polyphage. Polyphage was discovered by Hadley. Bacteriophage attacking E. coli are called as polyphage. What are the polyphages? The bacteriophage. What is bacteriophage? The virus which infects the bacteria is known as bacteriophage. The bacteriophage attack into the E. coli. E. coli means which is a gram-negative rod-shaped organism. E. coli, Escherichia coli. So bacteriophage attacking E. coli are called as polyphage. Polyphage are designated as T4Phages. This may be in your objective sessions. What is the other name of polyphages? The polyphages can be designated as T4Phages. Okay, they are numbered as T1, T2, T3. Okay, ma? T odd phases and T even phases. Okay, next ma? These are the most complex hexagonal head. Already we discussed hexagonal head will be there and rigid tail will be there and contractile fibers. This is the basic model of any bacteriophage. These are categorized into different bases. A is consist of head, tail phages. Example for this polyphages T2, T4, T6. These are T even phages. Coming to the B subgroup, they are similarly A, but they lack contractile sheath. Contractile sheath which helps for the attachment of the bacteriophage to the bacteria. They helps for the attachment of bacteria. So they are called as poly-even phages T1, T5. Next, coming to the C group, these are same as B type. They doesn't have any contractile fibers, but the tail has shortened than the head. Tail is shorter than the head. Head is longer and tail is shorter. Coming to the D group, they have head. Head is made up of large capsomes. Capsomes are made up of proteins, but not tail. The D subgroup of bacteriophages does not contain tail. Example for this 5X174, which you know in your intermediate. Next, coming to the E group. These are similar to D type. Instead of they are having small capsomes. Capsomes are the proteinaceous layer present in the bacteriophage. Example MS2. This is used in the replication mechanisms. Coming to the F, these are the filamentous bacteriophage. Examples polyphages. Filament-like structure will be there. Next, coming to the G group, they are lipid envelope. They consist of a lipid envelope surrounding to the nuclear membrane. So these are the groups of bacteriophages. Next, coming to the life cycle. Life cycle phases are the best understood in all the viruses. To know about the viruses, we have to study about this phases. Like that of the viruses, the bacteriophage is also undergoes two types of life cycles. That is, one is lytic cycle and other is lysogenic. First of all, we will discuss about the lytic cycle. The lytic cycle is a phase replicative cycle that accumulates the depth of the host cell. The lytic itself indicates lysis of the cell. So, the depth of the host cell, whatever the host it was attached with the contractile fibers, it kills the host cell and releases its the daughters. That is called as lytic cell. Lytic cell division means the host cell which is attacked by the bacteriophage was killed. That is called as lytic cycle. The lytic cycle produces new phages and lyses the host and releases in the progeny. The phages that are reproduced only by the lytic cycle are called virulent phages. Virulent phage means very harmful phages. The phages which are reproduced only by the lytic cycle. Lytic means, what do you mean by lytic cycle? Which kills the host cells and releases its progeny. They are called as lytic cycle. The phages which are released by these lytic cycle, they are very virulent phages. Virulent means harmful phages. Bacteria have defense against these phages. If any bacteria enters into the body, naturally our body reacts and defends against. If we are susceptible, that means sensitive, the bacteria will create the disease. At the same like this, when the virus attacks to the bacteria is also have defense against the phage. They consist of some of the restricted endonucleases that are called as restricted enzymes. These enzymes recognize and cut up, create the phage DNA. Okay. Next, I am coming to the life cycle. The first stage is penetration stage or ingestion. First of all, the tail fibers, we already studied in head, collar, tail. These are the three main parts, head, collar, tail. So, the tail fibers of the virus bends to bring the spikes and basal part to contact the bacterial cell wall. That means the virus comes and attaches to the bacterial cell wall. So it compresses itself. The sheath is present the viral genome injected into the bacterial cytoplasm. So it releases its nuclear material into the bacterial cytoplasm. The empty capsid remain outside of the host cell. That is called as host. This step is activates and acquire ATP. Next stage is elliptic stage. In this stage, the immunity against the infection by the same force by production of specific enzyme called as repressor. The bacteria is producing some of the restricted enzymes. Now, against this restricted enzyme, the fast produces some of the enzymes which are known as repressors. With this repressors suppresses the cellular immunity of the host. The immunity of the host will be suppressed. So the bacteria phage synthesis its protein coded by the bacterial DNA using the host amino acid pool. The protein synthesis is called as early proteins. Okay, now this is called as elliptic stage. Next, coming to the maturation stage or viral assembly. The head, tail are first assembled. Head, tail will be first assembled separately. Then they can be attached. First head will be formed, then tail will be formed. They get attached to the viral particle. The time period between injection of viral nucleic acid and first appearance of the phage is known as elliptic period. The time period between the injection of viral nucleic acid and the first appearance of the phage is known as eclipse period. The time period between injection of viral nucleic acid to rupture the host cell is known as latent period. Okay, ma? The final stage. The cell wall at this stage is also called as lysis and release of new virus. The cell wall of the host bursts out of the end and latent period and the phase is liberated. This phenomena is known as lysis. The number of variants produced by host is specific and termed as a burst cell. Generally, the size is 200 to 300 nanometers. Here, the whole cycle will be exhibited. You can see attachment, entry, and there the different parts will be synthesized. After that, we already studied assemble will be happened. Assemble and phase attachment. Head will be attachment, tail fibers will be attachment. Finally, the bursting of the cell. Cell will be released. This is the whole cycle. Okay. Attachment, entry of the DNA, entry of fast DNA, and degranulation of host DNA. It break down the host DNA and it synthesizes the viral genome and proteins. There it get assembled. Different parts can be prepared differently. After that, they will get assembled. Each part will be assembled. Then the phage assembly will be occurs. Then release of the phage will be occurs. Okay, this is called as lytic cycle. Lytic means the lysis of the cell. The bacteriophage comes and attaches to the bacteria. They use all the machinery of the bacteria. Finally, what is to it ruptures the cell wall of the bacteria. So it is called as lytic stage. Then, so coming to the lysogenic cycle, the lysogenic cycle replicates the phage genome without destroying the host cell. Lyso is communicate with the bacterial genome. It communicates with the bacterial genome. It can multiply along with the genome. So it reproduces and produce the bacterial genome and also the viral genome. Lysogenic, remember, it doesn't cause any harm to the host cell. That is called as lysogenic cycle. The lysogenic cycle replicate the phage genome without destroying the host. You can't destroy the host cell. The viral DNA is incorporated into the host cell chromosome. The viral, as usually, comes and attached, but it doesn't cause any harm. The viral DNA molecules incorporate into the host cell chromosome. This integrated viral DNA is known as prophase. What is prophase? The integrated viral DNA into the bacterial DNA is known as prophase. Every time the host divide, it copies the fast DNA and passes the copies of daughter cells. Number of daughter cells will be produced. Here the see the diagram, the fast as usually the fast get attached the DNA, the fast injects its DNA, the fast circulates and certain factors determine. So this is sometimes it may go for lytic cycle, sometimes it may go for lysogenic cycle. In case of lysogenic cycle, new forms of DNA and proteins are synthesized and assembly will be occurs, can circulate. Okay, ma? Next. So lytic cycle, the same thing, number of progeny will be produced. Here you can see the bacteria will be reproduced, copies of progenies transmitted into the daughter cells. Profast DNA, here the separate thing is the profast DNA integrates into the bacterial chromosome, becoming a profast. Profast means the fast which consists of the host DNA and its DNA is called as profast. So it goes and multiplies and giving a number of progeny. Next, coming to the significance of lysogenic cycle. Lysogenic fast has been shown to carry genes that can modify in the Salmonella O antigen. Salmonella typhi, as you might all know, which is a food and waterborne infection. It is a gram-negative organism. So next, it is also toxins produced by Corynebacterium diphtheria is mediated by gene carried by a beta fast. So significance of lysogenic cycle. There is a significance in the Clostridium botulinum, a positive agent of food poisoning, make several different toxins of which we actually encoded by progeny genome. Lysozyme bacteria are resistant to superficial by same or related phase. This is known as super infection immunity. Okay, this is the significance. Next, what are the phases? Why the phases are good for? These phases play an important role in ecology and evolution of bacteria. Phase assists identification, classification and detection of pathogenic bacteria. Phases are useful for whether the bacteria is pathogen or it is an avirulent strain. They are used in fast therapy. They are mainly used in the fast therapy. They eliminate pathogenic bacteria like Campylobacter and Listeria in fresh food to reduce the food poisoning. Listeria, which is present in the refrigerator, Listeria monocytogen, which causes abortions. The pregnant lady can eat the food from the spoiled food from the refrigerator. It causes abortions. By using fast, we can eliminate this problem to some extent. Next, the significance of bacteria, as we might already discussed, it is helps for the transformation of genetic material from one cell to other cell. Lysogenic cycle is useful for getting of new characters. So these are very important. The bacteriophages are very important. They liberate the more. They also helps for the development of monoclonal antibodies. Okay, this is about bacteriophages. So bacteriophages are very useful. You have to remember, bacteriophages are nothing but the viruses which eats the bacteria. There are two types of life cycle, lytic and lysogenic. Lytic cycle means which kills the host completely. They are virulent phages. Okay, polyphages is also called as T4Phages. Next, coming to the lysogenic cycle, here it replicates along with the host DNA, gives number of copies of the viral genomes. Okay, you remember now? Thank you all.